In this lecture, we're going to be talking about the basics of the nervous system. We'll talk about the cells that make up the nervous system, the general divisions of the nervous system, and if you want to learn more about details of brain areas and their functions, that's going to be in part 2 of this particular lecture. So let's get started. There are two major cell types in the nervous system, one of which is neurons. So you might have heard this called nerve cell, brain cell. In behavioral neuroscience or in any field of neuroscience, we call these neurons. They represent the workhorse or fundamental unit of the nervous system. However, this information is relatively recent. I guess when you think of all the scientific discoveries in the world, our understanding of the fact that the nervous system is made up of neurons is relatively recent because it was dependent on being able to see them using microscopes. Those microscopes that allow us to see neurons as separate entities weren't developed until the mid-20th century. Neurons are really, really cool because they allow communication between themselves. They have complex connections, and neurons in different areas of the brain look and are shaped differently because structure equals function. Prior to kind of being able to see neurons as individual entities, there were two scientists that were arguing what the fundamental unit of the nervous system was. The first one was Camillo Golgi. He was an Italian neuroscientist that came up with what was called the nerve net theory. He argued that the nervous system was one continuous network, like a web of neurons or web of cells, that they were all connected to each other. There was no separation. On the other hand, another scientist, a Spanish neuroscientist, Santiago Ramon y Cajal, who also was an incredible artist. So if you get a chance to look at any of his paintings, they're quite incredible. He used Camillo Golgi's stain to fill neurons completely, and here is actually an image of a filled neuron with the Golgi stain. And he drew these neurons meticulously, studying them under a microscope, again lending themselves to see some beautiful images. But he concluded, based on these drawings and investigations, that there was no network, that the nerve net theory wasn't correct, that in fact there were individual units called neurons. And sadly for Camillo Golgi, Ramon y Cajal ended up being correct, that neurons are in fact the single fundamental unit of the nervous system, and it is something that exists in neuroscience today, right? So it's something that he observed, and I should mention his observations and theory of the neuron doctrine was developed well before scientists were able to confirm that in fact neurons were individual entities. So let's talk about a neuron, what it is, what its parts are. This cartoon of the neuron is a general image or an average image of a typical neuron, but neurons definitely vary in what they look like. But we'll talk about the neuron parts using this cartoon image. So the most prominent structure of the neuron is the cell body or soma. So cell body and soma are one and the same thing. We call soma, it's a fancier term for cell body, but it means the same thing. It is kind of the biggest structure of the neuron, and it's typically where you find organelles, which are kind of the organs of the cells, residing. So you might have heard of nucleus, mitochondria. Those are organelles, and they are found largely within the neuron cell body. We'll talk about these organelles in a later slide. Coming off of the soma are two major structures, the dendrites, which are kind of creating a crown on the cell body. They represent the receiving portions of the neuron. So if there's another neuron that's going to communicate with this one, it's going to do so at the level of the dendrites. The other structure that comes off of the soma is the axon. The axon tends to be incredibly long, depending on the cell, and there's usually only one axon coming off of the cell body. A neuron can have anywhere from 1 to hundreds of dendrites, but all neurons typically have just one axon. It's this long cable-like structure, or fiber, because it's very, very thin, that carries what we'll learn about in later lectures called the nerve impulse or action potential. And so it carries this electrical impulse down the length of the axon all the way to its end. The part where this action potential or nerve impulse starts, or the beginning of the axon, is called the axon hillock. In addition to the axon hillock, you'll notice that some axons are covered with fatty material called myelin sheath. Just like the insulation of the wires, the electrical wires in your house, myelin sheath serves to insulate the axon to speed the movement of this nerve impulse down the length of the axon. And we'll learn all about it when we talk about action potentials in the coming weeks. But the action potential, or the nerve impulse, starts at the axon hillock. Its speed is regulated by the fact that there's myelin sheath covering it, so it insulates the action potential. But you'll notice that the entire axon isn't covered entirely with myelin, that there are bare parts of the axon showing up. Those are called nodes of Ranvier, and they also help in the propagation or the speed of the nerve impulse or action potential. When the action potential or the nerve impulse gets to the end, right, the end of axons are called terminals. It's the point at which the information is then sent to the next neuron. So if this neuron's dendrites had neurons communicating with it, you would see axon terminals kind of from other neurons coming very close to the dendrites or the branches of this neuron's dendrites. Lastly, as I mentioned, the axon terminal, they contain chemical messengers that are then used to communicate with other receiving neurons or dendrites of other neurons. So now let's talk about what can be found in the soma or cell body. So I don't want you to be overwhelmed by this because it's a lot of organelles, just like your body contains a lot of organs to keep you alive. The cell body or soma of a neuron, of any cell actually in your body, contains all of these organelles. You're probably familiar with the nucleus. It contains all of your cell's genetic material. So every single cell in your body has a nucleus, and it contains all of the genetic materials that you need that are kind of jam-packed into 23 pairs of chromosomes. Surrounding the nucleus, you'll see these kind of compartmentalized structures. There are two different types of one major compartmentalized structure that is very close to the neurons because it's involved in protein production, and that's the rough endoplasmic reticulum. So the rough endoplasmic reticulum almost has it looks like it's contained or following or continuing the nucleus, but it's really not. It's just allowing a pathway for information from the nucleus to pass into the rough endoplasmic reticulum. You'll see it's got little studs of stuff all over it. These are ribosomes. Ribosomes represent an organelle that make proteins, which are very, very important for the functioning of neurons, but also all cells of your body. Sitting outside of the rough endoplasmic reticulum is something called smooth endoplasmic reticulum. It's called smooth because it's not studded with ribosomes the way the rough endoplasmic reticulum is. This particular organelle is responsible for making things like fats and steroids. An example of a steroid is our hormones, estrogen and progesterone, but not all cells produce those steroid hormones. We're going to move here onto this side. We'll talk about the mitochondria. You may have learned about the mitochondria in introductory biology in high school. The mitochondria is the sole source of energy for the cell. It takes sugars like glucose, for example, and breaks it down into a usable form of energy called ATP through a really complicated process called the Krebs cycle. You don't need to worry about that, but the mitochondria is the only organelle that makes energy that's used by all of the organs, or all of the cells. The next structure, the next organelle we'll talk about is the Golgi apparatus. The proteins that are made in the rough endoplasmic reticulum then kind of fly through the cell, or I guess float through the cell. We'll talk about what it's floating through, and enters the Golgi apparatus. In the Golgi apparatus, these proteins are packaged and then kind of shuttled in these packages to wherever it is they need to go, wherever they're going to be used. What I failed to mention that's kind of important here is that the entire cell is covered by the cell membrane, and the cell membrane is really, really important in neuronal function. We'll talk about what consists of the cell membrane and what's in the cell membrane. So it's a fluid structure. But the cell membrane is very similar to your skin. It separates the internal components of the cell from the external or external environment. What I should also mention that's not in this particular diagram is the fact that the internal structure, or the inside of the cell, is completely filled with a fluid called cytoplasm. And again, these organelles are floating in the fluid. They're not like hanging out in air. The cytoplasm, which isn't mentioned here, allows movement of material between these organelles and through the cell. So these organelles are constantly moving around. They're not fixed in any particular location, as this image might lead you to believe. It's a fluid that's constantly moving, and so the organelles within the cytoplasm are moving as well. What's keeping or helping keep the shape of a neuron, especially given the fact that the neuron has a very specific shape, are proteins called cytoskeleton. They're called cytoskeletons because they serve the same function as the skeleton in your body. If you didn't have those bones in your body, you wouldn't be able to keep your body's shape. And so cyto means cell, and so cytoskeleton is the skeleton of the cell. These are proteins that some of which are very rigid, others are built and grow as needed. So there are some cytoskeletal proteins that are very dynamic, others that are much more rigid and firm. You don't need to worry about that, but you do need to know that cytoskeletal material is made of proteins, and it helps keep neurons its shape, or lends neurons its shape. Lysosomes are an organelle that help break materials down. So they contain other proteins, which are called enzymes, that break things down in the cell, that kind of digest them. So you can think of lysosomes as like the digestive system of a cell. So that's it for the organelles. We'll talk about some more than others. So we'll definitely be talking about the nucleus. We'll be talking about the ribosome, cytoskeleton, mitochondria, as well as Golgi apparatus in greater detail as we talk about proteins and protein production. So now let's talk about the different types of neurons. I kind of alluded to this, that neurons aren't all the same in terms of their shape, and different neurons actually have different shapes as well as extensions. So there are three major types of neurons based on the number of extensions that come off of the cell body or soma. We have unipolar, bipolar, and multipolar. So the term pole represents ends, and so what it's saying that in unipolar neurons there is just one end. There are no two ends. Bipolar has two ends. Multipolar has many ends. So let's talk about unipolar neurons. Unipolar neurons, the cell body or soma hangs off the extension, the single extension that comes off of, or that exists in a unipolar neuron. One end represents the dendrites, and the other end represents kind of the continuation of the axon. These neurons are very unique. They are only found in the sensory systems, such as your nose, your sense organ's nose, and I believe your skin. Most other sense organs are filled with bipolar neurons. Bipolar neurons have their cell body kind of smacked in the middle, and one end of the cell body represents dendrites, the other end represents axons. So both bipolar and unipolar neurons are found in sense organs of the nervous system of the body. Multipolar neurons are found everywhere and primarily are the type of neuron that you'll find in the spinal cord and the brain. It's called multipolar neuron because unlike bipolar, where there's one dendritic extension that then branches, multipolar neurons have multiple dendrites coming off the cell body and a single axon that then eventually branches when it gets closer to the end. So these neurons vary in their structure across these three types, but I do want to mention that multipolar, although they generally take on this shape, they are also varied in their structure, and largely they differ in the variety of dendritic branches that they create. The other type of cell within the nervous system, in addition to the neuron, is the glial cell, or glia, which is the plural form of glial cells. The astrocyte, which you see here, it kind of looks like a star, which is why it's called astrocyte. It looks like a star, and then it's got these extensions coming off the cell body, and then it's got these funky little flat structures at the ends of its extensions called end feet. We'll talk about what those end feet do in a couple of slides soon. But astrocytes are kind of jack of all trades. They do a lot in the nervous system. For example, they fill spaces between neurons, so in some ways they influence whether neurites, which are the baby dendrites, which is what baby dendrites are called, they influence whether neurites grow or retract. They also regulate chemical contents of the brain, so we'll learn about astrocytes sucking up some of the chemical messengers that neurons release to ensure that the balance of these chemicals doesn't disrupt brain function. They also engage in something called phagocytosis. Phagocytosis involves kind of gobbling up or breaking down dead matter. So I think of phagocytosis, and maybe I'm dating myself when I say this, but I think of phagocytosis as like the game Pac-Man, where they engulf whatever substance needs to be broken down and quote unquote digest or break those substances down. So astrocytes participate in phagocytosis. So if there's dead tissue or dead matter or quote unquote trash in the brain, astrocytes will go to it and gobble it up. They play a really important role following head injury or brain damage in any way. Astrocytes, again, are moving through the brain. They can multiply, even in the adult brain, and they will go to the site where the damage has occurred and create kind of like a scab over the damaged area, much like you get a scab over a cut or an injury on your skin, and keep that area contained. While they're creating the scab, they're also engaging in phagocytosis. The other type of glial cell, and there's two of them in this category, they're called the myelinating glia. So I mentioned to you earlier in an earlier slide that axons are covered with a fatty substance called myelin. That myelin is made by glial cells, and there's two different types of glial cells. You can see one type of glial cell here. This is called the oligodendrocyte, and you can see the other type here. It's called the Schwann cell. So oligodendrocytes have one cell body, and they reach out and can wrap multiple axons at one time. Schwann cells, only one Schwann cell can wrap one axon at a time. Both of them are producing myelin, which is that myelin sheath that's covering the axon to help insulate it. In between the wrapping of myelin sheath, where there's the bare axon, that's called the node of Ranvier that we talked about in the earlier slide. In addition to the number of axons oligodendrocytes and Schwann cells can myelinate, they also differ in where they are found in the nervous system. Oligodendrocytes are found in what's called the central nervous system, and we'll talk more about that in the coming slides, but it's made up of the brain and the spinal cord. And Schwann cells are found in the peripheral nervous system, which is pretty much everything else. Like all the nerves in your body belong to the peripheral nervous system. So here's a better image of oligodendrocytes and Schwann cells. Here's an oligodendrocyte, and you can see it's got these extensions that's myelinating several neurons at any one, or several axons at any one time. Here in the peripheral nervous system, you're going to find Schwann cells wrapping themselves around the axons of the neurons that form the peripheral nervous system. It's really important to kind of study these glial cells because they get attacked in a disease called multiple sclerosis, or MS. You may have heard of MS. MS is an autoimmune disease, and scientists still don't understand why it occurs. But what ends up happening is that your body's immune system essentially starts attacking the myelin on the axon of the peripheral nervous system as well as the brain. And when the myelin is damaged, the speed of that nerve impulse then is weakened, right? And so people end up not being able to perform functions well. You start seeing damage first in the neurons that control motor function. And so typically people who have early stages of MS will see problems associated with their motor function. Those are the ones that are most visible. But people with MS may also struggle with thinking because your neurons are part of your brain, and the myelin on those neurons are also getting attacked or damaged in MS. So scientists are actively trying to figure out how we can get neurons or myelin to be reformed in MS or slow down the breakdown. The two other glial cells that we'll talk about, or that you're going to be introduced to, are radioglia and microglia. Radioglia are not found in the adult brain. They only exist in the developing brain, and they help neurons migrate from one location to another. When we talk about development in this course, you'll learn more about radioglial cells and their shape. Microglia, and this is a microglial cell here, serves as the immune system of the brain. They tend to attack any invading microorganisms that may be present in the brain. They also, like astrocytes, participate in phagocytosis. So they are much smaller, but there's lots of them. So I should mention that there are way more glial cells in the nervous system than are neurons. And glial cells, while they aren't the ones responsible for transmitting information, in the absence of glial cells, the neurons would not function as well as they need to. So they are definitely an important support system for neurons. An important feature of glial cells is something called the blood-brain barrier. So astrocytes participate in the blood-brain barrier, but it's an important feature of the nervous system. So there exists a physical barrier between the brain and the rest of the body. And when I mean the rest of the body, I mean blood vessels, right? Because blood vessels go everywhere, but the blood vessels in the brain are different than the blood vessels in our body. So in our body, our blood vessels have giant gaping holes that allow material to flow out of the blood vessel and into the blood vessels. But this is not true for the capillaries, which is a type of blood vessel, very, very small, that exists in the brain. There are absolutely no gaps. In fact, the only way that materials can get through blood vessels in the brain is through diffusion, which is it's going to have to easily pass through the blood vessels, like gases, oxygen and carbon dioxide easily pass through the blood vessel, or, as is the case for most other substances, they have to be actively transported from the blood vessel into the brain or the nervous system. So what you'll see in the blood vessels, so blood vessels are made up of endothelial cells. It's a type of cell that lines or creates the blood vessels. In blood vessels of the body, you'll see huge spaces between the blood vessels, the capillaries, but in the endothelial cells of the capillaries of the brain, there are tight junctions, which are creating kind of this immovable space. There's no space. Like it's a kind of like a barrier, kind of a wall between the contents of the blood vessel and the rest of the brain. In addition to the blood vessels type contributing to the blood-brain barrier, you have astrocyte end feet. So remember in the prior slide, I showed you that astrocytes have these like flat structures at the ends of their extensions. That's what these end feet are doing. One of the things that these end feet do is they kind of line the outside of the capillaries, preventing material from going or getting from the capillaries to neurons. So these astrocytes serve as kind of like the next or part 2 barrier between the contents of the blood and neurons. So they serve as a physical barrier between the blood vessels and neurons. They're also important. So they take, right, there are nutrients that come in the blood. They take the nutrients that come through the blood and then transport them to neurons. So they serve as a really important component of the blood-brain barrier. And you're probably wondering, like, why does the blood-brain barrier exist? Well, if materials can easily be transported between the blood and the brain, the really delicate chemical balance that is needed to be maintained in the brain would not occur, and as a result, your brain wouldn't be able to function. So in later lectures, you'll learn about the fact that the neurons have to maintain a particular balance of chemicals both in and outside of the cell. And if we were to allow all of the materials from the blood to enter the nervous system, that balance, right? You eat a heavy, salty meal, those chemicals can easily get into the brain, and then you're going to risk altered brain function, which nobody wants. It's important to mention that not all areas of the nervous system have a blood-brain barrier. There's an area in the brainstem called the area postrema, which is also known as the vomiting center. Sorry for the lack of a better word, but it tells you what it does. If we ingest a chemical that is really bad for our body, that could be toxic, it is going to activate, right? Those chemicals are going to enter the area postrema and trigger the vomiting reflex so that we can expunge or get rid of any chemical that could potentially harm our body. This is also true when we experience, when we consume something that is not good for us, that might have a pathogen. It's going to trigger activation of the area postrema, and we're going to do our best to get rid of all of that harmful substance that we purposefully or accidentally ingested. All right, now we're going to be moving on to general kind of parts of the nervous system. Now that we've talked about the cells of the nervous system, we can break the nervous system into 2 major divisions, the central nervous system and the peripheral nervous system. So these 2 are the 2 major divisions of the nervous system. The central nervous system, which we'll spend a lot more time in part 2, part 2 of this lecture, the central nervous system is broken down into the brain and the spinal cord. The peripheral nervous system has largely a functional division. So we have the somatic and the autonomic nervous system. Somatic nervous system is more, I would say, voluntary. The autonomic nervous system is way more automatic. Like, you have absolutely no control over this. The autonomic nervous system is further subdivided into the sympathetic and parasympathetic divisions. The PNS and the CNS, the peripheral nervous system and the central nervous system, in addition to having different types of glial cells, they also are unique in the fact that axons of the peripheral nervous system can regrow and regenerate following an injury. The axons of the central nervous system, once they are damaged, do not recover from injury. So if you end up with, brain damage where axons are severed or cut, as is the case with concussions, those axons are not going to be able to recover. Part of the reason is that the central nervous system produces what are called inhibitory molecules that prevents regrowth of the axons of the damaged neuron. There are no inhibitory molecules in the peripheral nervous system, and so the axon is able to regrow in the peripheral nervous system, unlike the central nervous system. So they differ in that way as well. I do want to spend a little bit of time talking about nerves. So nerves, even though they seem pretty small, they are actually made up of hundreds of thousands of axon bundles. Nerves are carrying information to and from the central nervous system and your body. So they are kind of the messengers of information to and from your body and the central nervous system. There are two different types of nerves. Efferent neurons, so they're effecting muscles, they're carrying information from the central nervous system to muscles of the body, and these tend to be motor in function, so they control the muscles of your body, they do things, but they also control things like digestion. Afferent, these are nerves that are carrying information from the skin and sense organs to the central nervous system. These tend to be sensory in nature because they're carrying sensory information to the central nervous system. So what you're looking at here is kind of like a slice through the spinal cord, and there are two parts of the spinal cord. So the inside of the spinal cord looks like a butterfly. The top half of the spinal cord handles sensory information. The bottom half of the spinal cord handles motor function. So sometimes the nerves carry or have both efferent and afferent nerve fibers. So they are a mix of nerve fibers carrying both to and from the central nervous system. Here, again, it's not color-coded in your body, but in pink here are the nerve fibers carrying sensory information. In blue here are the nerve fibers carrying motor information. So these pink fibers are afferent nerves. The blue fibers are efferent nerves. Okay? Don't worry about all these other terms, but these roots that are coming off of the spinal cord join together to form a single single nerve coming off of the spinal cord. It's important for these efferent and afferent nerve fibers to work together or coordinate. The afferent nerve fibers detect stimuli in our sense organs, and these fibers carry information to the central nervous system. The central nervous system then decides what to do with that information and transmits the motor response through the efferent fibers to control muscles of your body, so then you can react appropriately to the stimulus. The only type of stimulus that does not involve the brain is a reflex. Reflexes only occur in the spinal cord, but they occur due to communication between sensory fibers and motor fibers. So the sensory information communicates directly with the neurons that control motor function in the spinal cord. Okay, peripheral nervous system, right? We spent a little bit of time talking about nerves and nerve fibers and what they are. The peripheral nervous system is any portion of the nervous system that's not contained within a bone. So here, anything in blue, which are all the nerves of your body, belong in the peripheral nervous system. You know you're dealing with the peripheral nervous system compared to the central nervous system because the central nervous system is encased or contained within bone. The brain is contained within the skull, and your spinal cord is contained within the vertebral column. The nerves are structurally, that is physically, divided into two categories: cranial nerves, which are nerves that come out of your brain, and spinal nerves, which are nerves that are coming out of your spinal cord. Functionally, the peripheral nervous system, as I've mentioned to you already, is divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system controls all of the muscles of your body. They're also the ones that are carrying information of from your sense organs into your brain. The autonomic nervous system handles automatic functions, and it has that additional two divisions of sympathetic and parasympathetic. So just to give you an idea, here is the spinal cord encased in the vertebral column. You can see based on the division, the spinal nerves are broken up into different categories. So if you've ever had any problems with, squeezed or compressed nerves, you might have been told that, it's your T4 or your L5. These nerve fibers or these nerves are targeting specific areas of the body. And so scientists, or I should say doctors, based on what parts of your body have lost sensation or mobility, can determine what area of your spinal cord is damaged or what nerve fiber is being compressed. So these are all the spinal nerves. Here are all the cranial nerves. There are a total of 12 cranial nerves. These are nerves that come off of your brain. That's why they're called cranial nerves, because they're coming off of the brain. And all of these nerves belong to the peripheral nervous system. But you can see the cranial nerves are largely handling functions that exist within the head and neck, with the exception of one, which is the vagus nerve, and that's the one that controls things like respiration and digestion. All right, you don't need to know kind of the details of the cranial nerves or what they are, but I wanted you to see what I meant by cranial nerves as well as spinal nerves. Oh, an interesting fact, the spinal cord actually ends here, and the rest of your spinal column is actually just fibers, nerve fibers. They are not filled with spinal cord material. Most people think their spinal cord goes all the way down to the base of their vertebral column. That is not true. Your spinal cord actually ends kind of the middle of your back. Okay, what is the somatic nervous system? So the somatic nervous system carries information from the sense organs and gathers it and sends it to the central nervous system. It also controls the muscles of your body, your skeletal muscles. This represents the voluntary portion of the peripheral nervous system, and I know you don't have control over what's coming through your sense organs, but you do have control over the skeletal muscles, which is why it is called the voluntary portion of the peripheral nervous system. The autonomic nervous system is purely involuntary. You have no control over this, and it's responsible for controlling things like smooth muscle, cardiac muscles, and glands. So you have no control. It's involuntary. There are two divisions of the autonomic nervous system. There's the sympathetic and the parasympathetic. Both of these divisions speak to or control the same organs of your body. And they have two functionally different roles. The sympathetic nervous system is responsible for expending energy. it helps break down stored energy, preparing us for a lot of movement and energy burning. It governs what's known as the fight or flight response, or the stress response. The parasympathetic nervous system, which opposes the sympathetic nervous system, is responsible for conserving energy. So it brings the body back to rest, to homeostasis. And so you'll see here, these represent the sympathetic division, the parasympathetic division, and both the sympathetic and parasympathetic divisions speak to exactly the same organs. They are typically not activated at the same time. So when the sympathetic division is activated, typically the parasympathetic division is shut down, and the opposite is true when the parasympathetic division is activated. Most of the time we are in the parasympathetic division is activated unless we are feeling stressed or worried or anxious about something, in which case the sympathetic division will be activated. And so that's it for this particular lecture. In this lecture, we talked about what neurons were, as well as the internal organs. We talked about glial cells and the different types of glial cells, especially astrocytes that are important for maintaining the blood-brain barrier. We talked about the two major divisions of the nervous system, central and peripheral. And we spent some time talking about the peripheral nervous system. Towards the end of this lecture, we'll spend a lot more time in the central nervous system in part 2 of this lecture.